Without further ado, Alicia, welcome, and feel free to present.
Great, thanks. So yeah, as you mentioned, I'm a postdoc in Mark Daly's lab, and this is sort of a continuation of my work as a grad student in Carlos Bustamante's lab. But the last part of it, which I think is probably the most important part on genetic risk prediction, all originated here. So just to provide some motivation, as you have probably heard, there's this massive European study bias in genetic association studies. So for example, David Goldstein's group put out a study in 2009 showing that 96% of participants in genome-wide association studies are European descent. While that's increased lately, the other populations primarily contributing are of East Asian descent. So the vast majority of US minorities are not contributing to genetic association studies. So this leaves a large amount of genetic and phenotypic diversity yet undiscovered. A large project here at the Broad is the Exome Aggregation Consortium, and this has also shown that European populations have the fewest homozygous loss of function variants, which are a very important type of variant to study for disentangling a disease role. So if you wanted to choose probably the worst population to study to understand this role, you might choose Europeans. So today I'm going to talk about just a few things. So here's like the outline generally. So I'm first going to start with general population structure, then talking about GWAS tools and some study biases, and then end on probably the most important part of the study, which is the transferability of GWAS through polygenic risk scores, which I'll define later. So the primary data set that we used in this project was 1,000 genomes phase 3 data set. So as you can see on this map, the 1,000 genomes data consists of about 100 samples from populations sampled all throughout the world. In the bioRxiv preprint, you can go online and learn more about local ancestry, principal components analysis, when admixture started for a lot of the populations in the Americas. So the ASW and ACB are African American populations from the Southwest US and the Caribbean. And then the Puerto Ricans, Mexicans, Colombians, and Peruvians are all Hispanic Latino populations that I focused on to better understand their admixture history in the preprint that you can go online and read more about. So what I really want to focus on in this talk, however, is the biases in genetic discoveries. So on the left, you can see a pie chart showing the global populations across continental groups. So obviously there are a lot of different colors here representing different continental groups, African populations, Latino populations, East Asian, Middle Eastern, European, Oceanian, and South Asian populations primarily comprise what we know as the 7 billion people walking around the earth today. However, if you look at what's been studied primarily in genetic association studies, I'm picking on the Psychiatric Genetics Consortium just a little bit, but I think this is a really great consortia that's made major progress in psychiatric genetics and has studied schizophrenia, bipolar disorder, major depressive disorder, ADHD, and other diseases. What you can see by looking at the genetic makeup and the genetic ancestry of the participants in these studies is that they are, by and large, of European descent, with a small fraction coming from East Asian populations and essentially no samples coming from this, majority of the globe that we're not studying yet. So if we start to quantify what these biases do to our understanding of genetics through genome-wide association studies, we can look at something called heterozygosity. So on the y-axis of these plots is 2PQ. This is just a straight metric of heterozygosity. In the top chart, I'm showing you the whole genome. This red bar shows a West African population, the Yoruba. The blue bar shows you a European population, and the green bar shows you 2 East Asian populations. So as you can see, heterozygosity is the greatest across the whole genome in African populations, and the bottleneck out of Africa led to a decrease in heterozygosity in out of African populations. If you look at the bottom part, if you look at the genome-wide association catalog sites specifically, what you can see is that this trend is not reflected, and in fact, it looks like heterozygosity is the greatest in European populations, again reflecting this study bias. If we go forward then into admixed populations, we can break down their ancestry into the ancestral origins that created these populations. So we can look at African tracts, we can look at European tracts, and we can look at native tracts across African American populations and across Hispanic Latino populations. And what you can see is that these biases are recapitulated within each of these ancestry tracts. So these biases comprise these admixed populations. So on the top, looking across the whole genome, again, you can see that heterozygosity is greatest in the African tracts, less in the European tracts, and even less in the native tracts than in the East Asian tracts, because of how the peopling of the globe occurred when, you know, Native Americans crossed the Bering Strait. There was another bottleneck. If you look at the GWAS catalog sites on the bottom part of this plot, you can see again that European tracts show the greatest heterozygosity, reflecting this study bias. So this leads to a question. We've quantified these biases, and we're curious about how these biases actually impact our ability to learn more from genome-wide association study findings in other populations. In other words, given that we want to transfer knowledge from genome-wide association studies in European populations, how well or poorly are we able to do this in populations of non-European descent? So the way that I did this is using polygenic risk scores. So polygenic risk scores really originated at the dawn of the GWAS era, when we were so sample size limited that very few findings were emerging from these studies. And so a primary goal was to try to get a grasp on how many signals would potentially be found in the future from genome-wide association studies when we were well powered with decent sample sizes. And even today, we're finding that we have way more signals to be discovered than are, you know, readily captured in schizophrenia, for example, with 70,000 samples. So the way that a polygenic risk score works essentially is by summing across the genome those independent signals that are contributing to this particular trait. So for example, I might take genome-wide association summary statistics, and if you look at this little Manhattan plot on the bottom part of the slide, you can see that I've just put a rectangle over the top part of the Manhattan plot, but looked at things that are more permissive than at this genome-wide association significance threshold of 5 × 10⁻⁸. So in constructing the polygenic risk score, you simply use summary statistics from a GWAS that's been done before, and take the effect sizes from that and multiply them by your genotype in your study and sum them across the genome. So there are a few things that you have to consider. One is linkage disequilibrium structure. So as you can see in many different genome-wide association studies in these Manhattan plots, typically a sure sign that you're capturing real signal and not just finding technical artifacts is that there's a peak with many different variants all leading up to that peak. So in order to not capture a redundant signal, you might thin for LD. You might also apply some p-value threshold so that you're looking at things that are more permissive than the genome-wide significance threshold, but things that are contributing more to the total heritable variation that can be explained by this trait. Okay, so in constructing polygenic risk scores, I'm going to show you some empirical data that I constructed using the 1000 Genomes project. So the first polygenic risk score that I constructed was for height, and you can see on this x-axis just some sort of arbitrary units, but greater polygenic risk score indicates taller height. And you can see here that this Northern European group looks like it's expected to be taller than the Southern European group, which is great. Maybe that reflects some adaptive event in Europeans is, the initial thought that we had. However, if you look across the global set of populations, what you see is that Europeans appear to be the tallest, followed by American populations, then South Asian populations, then African populations, and the shortest group of populations here is expected to be East Asians. Aside from pygmy populations in Africa, their African populations that are captured in the 1000 Genomes Project are expected to be as tall on average as the European populations, indicating that our polygenic risk scores here might be biased. I also looked at a trait, type 2 diabetes, that's been studied both in Europeans and using a multi-ethnic cohort. So taking the summary statistics from both of these types of cohorts, I constructed polygenic risk scores across the 1000 Genomes Project. And what you can see here is that the order of the populations changes depending on where the summary statistics came from, indicating a role for demography in these biases. So I just want to lay out some intuition for how we thought about this when we very first started and we're trying to get a grasp on this, which we later learned was not right. So if you had a representative population that's been reasonably well studied in a genome-wide association study, and you tried to construct a polygenic risk score, what we thought we would expect was this red line with a representative, normal curve of polygenic risk. If you had an understudied population, you might expect that you're explaining less of the variation in that particular population, and that your variance in polygenic risk is shrunk, reflected by this blue curve. Conversely, if you had an overrepresented study, perhaps you're explaining more of the variation than you did originally, and you might have something like this green curve. And then perhaps if you had a trait that was positively selected, like is known to have been the case in height in Northern Europe, maybe there would be these mean shifts in the polygenic risk score. But what we've shown empirically is that there are actually considerable biases simply arising from drift. So we wanted to get more of a handle on how this was occurring neutrally. And so we set out on a simulation study. So the background for our simulation study is that there was a demographic model in phase 1 of the 1,000 Genomes project, where using site frequency spectra for an African population, a European population, and a couple East Asian populations, they estimated some demographic parameters like the effective population size for each of these populations, some bottlenecking events and the timing of those events, and then the migration rates between each of these pairs of populations. So this is a relatively realistic demographic model that I used in all of the simulation studies going forward. So just to outline the demographic simulation setup, the very first step was to simulate genetic data for African, East Asian, and European populations. Since African populations have greater effective population size, there were more variance in general in the African population. I next assigned evenly spaced causal effect sizes globally, simply following a normal distribution centered at mean 0, with some variance based on the heritability and the number of causal markers. I then computed the true polygenic risk score for all individuals across all populations. Then I ran a biased GWAS to be as realistic as possible and reflect what happens when people do genome-wide association studies, specifically in Europeans. And then lastly, I use these European genome-wide association study summary statistics to compute some inferred polygenic risk scores, and then compared this to the true polygenic risk scores that are actually causal, and use this to assess prediction accuracy. So the first thing that we confirmed, which we were glad to see in our simulation setup that was fairly realistic and fairly complex, is that our mean true polygenic risk score across 500 replicates, so pulling from the Coalescent tree 500 times, reflecting something like 500 different traits, like one for height, one for type 2 diabetes. What we see here is that the true polygenic risk score is not significantly different across populations. They all have essentially the same mean of 0. However, if we look at our inferred polygenic risk scores from running a biased GWAS specifically in European populations, what we can see is that there are both mean and variance shifts. So the variance shift is quite a bit wider in European populations, reflecting the fact that we're explaining considerably more of the variation in European populations. We're explaining less of the variation in East Asian populations and the least variation in African populations. If we look across different replicates from the simulation, so as I mentioned before, this is reflective of a few different traits. So you could think of this as representing something like height and type 2 diabetes and then another complex trait. What we see from each of these independent draws is that the population order between the true and inferred polygenic risk score are unpredictable. And these biases are inconsistent across populations, which indicates that we're not able to use straightforward metrics and covariate, something like a principal components analysis that's used in genome-wide association studies to correct for population structure. Those methods would not work very well here. So we actually need to develop new methods to correct these population biases and polygenic risk scores across populations to get rid of this unbiased, or to get rid of this bias across these populations. Okay, so lastly, just looking at prediction accuracy, if we compare the true and inferred polygenic risk scores across populations, you might expect, based on the plots that I showed you before. On the left, you can see that we have the highest correlation between true and inferred polygenic risk scores in European populations, the second highest in East Asians, and we do the worst in African populations on the left. On the right, I broke down these polygenic risk scores into deciles, and then I looked at the log odds ratio of being a case compared to a control. And what you can see is that you do the best at distinguishing cases from controls in European populations across these polygenic risk scores, and we do considerably worse in East Asian and African populations. So this again recapitulates this point that prediction accuracy is highest in European discovery cohorts. So just to conclude, there are GWAS tools such as imputation accuracy, study participant fractions, array backbones, and all these sorts of things. All of these are biased towards European populations. The most important take home from this is that polygenic risk scores are unpredictably biased across populations. This is driven by allele frequency and linkage disequilibrium differences, and this is not straightforward to simply correct using principal components alone, like is done in linear mixed models or in standard genome-wide association studies. This difference in prediction accuracy is reflective of the fact that we're explaining less of the phenotypic variation, so lower heritability and lower polygenic risk score correlations in other populations. So I know that was super fast. With that, I want to thank my advisors, Carlos Bustamante at Stanford, Mark Daly here at the Broad and MGH, some co-conspirators, Emer Kenny for leading the project, or for being the senior author on this project. If you have any questions or comments, feel free to email me, and the bioRxiv article is up in case you want to check out more detail there. So thanks, guys.